Good afternoon, dear friends. In last lecture, I have discussed the different factors that are influencing on the disinfection. In that, I have seen the different factors. First, the number and location of microorganism. Then, second, innate resistance of microorganism. Then, third, concentration and potency of disinfectants. Then, physical factors. Then, chemical factors. In that, first, temperature. Then, relative humidity, water hardness, then organic and inorganic matter, duration of exposure, and biofilms. Today we will see the next one, that is evaluation of bacteriostatics and bactericidal action. There are different methods that are used for evaluation. First method, tube dilution and agar plate method. Then second, cup plate, filter paper and cylinder plate method. Then third, ditch plate or giant colony method. Then CAS test, capital KS, that is Kelsey Sykes test. Then in use dilution test. Then phenol coefficient test or Rydell Walker test, that is capital RW test. Then Cheek Martin test. These are the 7 tests that are used for evaluation. We will see one by one. First, tube dilution and the agar plate method. See here, the chemical. See, in this method, the chemical disinfecting agent is introduced into the agar medium. Yes, in this method, the chemical disinfecting agents is introduced into the agar medium or the nutrient broth medium. After that the medium are inoculated with the test microorganisms. The test tubes containing medium and the microorganisms are incubated at the test tube containing microorganisms. The test tube containing medium and the microorganisms are incubated at 30 to 35 degrees Celsius for 2 to 3 days. And then the results are observed in the form of turbidities or colonies. Here we can see the turbidities or colonies in different test tubes. See here, these turbid colonies are recorded and the activity of given disinfectant is compared as shown. In that, see here, first test tube it shows clear, then second it is slightly turbid, then third it is moderately turbid, and last that is more turbidity. It means the last test tube it shows higher turbidity, that is more turbidity. In agar plate method, first plate it shows absence of colonies, that is it means there is no colonies. Then second plate it shows approximately 100 colonies. Then third plate it shows approximately 500 colonies, and last it shows approximately 1000 colonies. So this is all about agar plate and the tube dilution method. Second method, that is cup plate, filter paper and cylinder plate method. In this method, heating is performed for melting the agar. In this method, heating is performed for melting the agar and cooled up to 45 degrees Celsius. The agar medium is inoculated with the, in this method, the agar medium is inoculated with the test microorganism and poured into sterile petri dish. In the couplet method, when the inoculated agar has been solidified, and the holes of about 9 mm in diameter are made by cutting the medium with sterile cork borer, and the antimicrobial agents are placed into these holes. See here, cup, plate and cylindrical method. In the filter paper and cylindrical plate method, the antimicrobial agents are applied on the surface of solidified inoculated agar medium by using the filter paper and the cylindrical or the cylinder respectively. On the incubation at the temperature of 30 to 35 degrees Celsius for 2 to 3 days, the zone of inhibition is observed. So here we can see the zone of inhibition. And then the diameter of this zone gives an indication of the relative activities of different antimicrobial substances against the test microorganism. Here there is different zone of inhibitions. Then third method, that is ditch plate or the giant colony method. A ditch is prepared in this method. The ditch is prepared in agar plate and the solution of antimicrobial substance is carefully poured. In that first ditch is prepared and then the solution of antimicrobial agent is poured. Then on the agar surface, a loopful of each test microorganism is streaked onward from the ditch. Then microbes resistant to the antimicrobial agent grows on right of ditch, whereas susceptible microorganism shows a zone of inhibition adjacent to the ditch or the center of plate. Then the width of zone of inhibition, it indicates the relative activity of antimicrobial substances against various microbes used during this test. See here, ditch plate method. Then third, that is calcicyc test, that is capital CAS test. In this test, the test is measured of the capacity of disinfectant to retain its activity when it is used repeatedly. When it is used repeatedly in different microbiological operations, then it is called as capacity test. Then in this test, the standard microbes like E. coli, Staphylococcus aureus are added into this disinfectant in 3 continuous lots at a time difference, that is 0, 10 and 20 minutes. Then these 3 lots are kept in contact with disinfectant for 8 minutes and sample are transferred at the difference of 18 and 28 minutes. See here the flowchart of this test. Then thus the efficiency of disinfectant is determined by its ability to kill bacteria and not by comparing with phenol. Then this test is performed both in dirty as well as clean conditions. Therefore, it also measures the effectiveness of disinfectant in presence of an organic matter. Then the selection of the test organism, that is Staphylococcus aureus, Pseudomonas aeruginosa, vulgaris and E. coli depend upon the type of disinfectant used for testing procedure. Then for carrying out the test under clean conditions, the dilutions of the disinfectant are made in hard water, whereas to run the test under dirty condition, yeast suspension is used for making the dilution of the disinfectant. Then the organism with yeast alone is added at time interval of 0, 10 and 20 minutes, and the test organism and the disinfectant are kept in contact for 8 minutes. Three set of 5 replicate culture are made in corresponding to each condition and rare, and these are incubated at 32 degrees Celsius for 48 hours, and growth is assessed by turbidity. Then evaluation of the disinfectant is performed on the basis of ability the ability of disinfectant to kill microbes or the lack of it. The result obtained after this are regarded as a fail or pass. Then the results will be negative if two or more negative cultures are obtained. Results will be negative if two or more negative cultures are obtained in a single set. In case of negative result, the disinfectant passes the dilution test, and after the first and second condition. Then third condition is not considered either fail or pass, but the positive culture serve as inbuilt control. A lower concentration of the disinfectant may be tested if the negative results are obtained after the third condition. So see here, this is the flowchart for this test. Next, that is in-use dilution test. In-use dilution test. This test is used for examining the quantity of viable organism. Yes, this test is used for quantity of viable organisms and also for testing the liquid disinfectant is determined on the basis this test is used for examining the quantity of viable organism and also for testing the liquid disinfectant used in hospitals. Then the efficiency of a disinfectant then the efficiency of disinfectant is determined on the basis of its capacity to kill the known number of standard strain of pathogenic staphylococcus within the given time and the surface. Then result obtained from Riedel-Walker test and its modification. Then in this test, the disinfectants are used in diluted form. In this test, these disinfectants are used in diluted form, and the dilution at which the efficacy and the potency of the disinfecting agent is determined. Then the current standard for in-use tests are provided by the capital AOAC. It means American Official Analytical Chemist, that mentioned the use of 3 bacterial strains, that is Staphylococcus aureus, Salmonella chloriosis and Pseudomonas aeruginosa. The standardized culture of the test organism are grown in the liquid media. The standardized culture of the test organism is grown in liquid media, and this media is standardized and metal carrier rings are dipped into it and removed and dried at 37 degrees Celsius for shown time. Then after that, the dried culture are placed into disinfectant. After that, the dried culture is shown in. After that, dry culture are placed in disinfectant solution made at concentration specified by the manufacturer. The culture is left for 10 minutes at 20 degrees Celsius. The next step, the carrier ring are transferred to a culture medium that allows the growth of any surviving organism. The number of organisms in the resulting culture is calculated, showing the effectiveness of the disinfectant. So this is all about this test. Next, that is phenol coefficient test, that is Riedel-Walker test. In this test, The suspension with similar quantity of organism is used in the estimation of efficiency and the action of different concentration of phenol and the disinfectant to be tested. Then a solution of the test disinfectant that sterilizes the solution of test disinfectant that sterilizes the suspension in a given time is divided by the corresponding dilutions of phenol. This gives the phenol coefficient. However, this phenol coefficient is not the measurement of practical functioning of the test disinfectant in the presence of organic matter. Then by Radel-Walker test, the phenol coefficient of the disinfectant may be calculated. By using this testing disinfectant and phenol, the different dilutions are prepared and 5 ml of each dilutions are inoculated. And the 5 ml of each dilutions are inoculated with 0.5 ml of the 24 hours broth culture of the organism. Then all incubated tubes, that is disinfectant, organism and phenol plus organism, are kept at temperature of 17.5 degrees Celsius in a water bath. Then subculture of each reaction mixture is prepared and transferred into 5 ml of sterile broth at a time interval of 2.5. See here, we can see in table also 2.5, 5, 7.5 and 10 minutes. Then the broth tubes are incubated at temperature of 37 degrees Celsius for 48 to 72 hours and examined for the presence of or the absence of the growth of microorganism. Here we can see the different dilutions and the time interval for this organism. The Radel-Walker test coefficient, it is calculated by using formula, that is the dilution of the disinfectant killing in 10 minutes but not in 5 minutes divided by the dilution of phenol killing in 10 minutes but not in 5 minutes. So this is the formula for calculating this coefficient. So it is conclusion for this test. If the Radel-Walker or the phenol coefficient for this test, for the given test disinfectant is 1, the disinfectant has same effectiveness as the phenol. Then second, if the phenol coefficient of the test disinfectant is 20, the disinfectant is 20 times more active than phenol. Then if phenol coefficient of the test disinfectant is less than 1, then it is less effective, and it is more than 1. If it is more than 1, it is more effective as compared to phenol. So this is the conclusion for the test. Then second and last, that is Cheek-Martin test. The phenol coefficient test is performed on laboratory level to study the bacterial action of compound with respect to phenol. Then in 1908, the Cheek and Martin suggested that the disinfectants are essential to act the presence of organic matter. So they recommended the use of dried human feces in the test system. Then as a substitute to human feces, the Garrow suggested to use a dried yeast. In this test, Salmonella typhi, that is your test organism, is inoculated into solution having a graded concentration of test substance, that is or the phenol in case of standard, and along with dried yeast. Then the components are left for 30 minutes at 20 degrees Celsius to interact and lastly duplicate. A subculture are made into nutrient broth. Then these subculture tubes are incubated for 48 degrees Celsius at 48 hours for 37 degrees Celsius, and observations are made regarding the presence of or the absence of growth of microorganism. and the phenol concentration that prevent the growth in the both system is determined, and then mean value is calculated. Then the same value will be obtained for unknown, and for calculating the coefficient, the value which are obtained for the value obtained for phenol is divided by the value obtained, or the value found for the unknown. So this is all about this all evaluation test of the, or the evaluation of bacteriostatic and the bactericidal action. I'll stop here. Next part we will see in next lecture.